Okay, yeah. So good day, everyone. Welcome to the first ever webinar hosted by the Mediterranean Neuroscience Society and Brain Organoid and Systems Neuroscience Journal. My name is Nermeen Zakaria, and I'm a council member for the Mediterranean Neuroscience Society. Today, I'm very delighted to be your moderator for this event. Today marks a significant milestone for our society as we board on this new journey of knowledge sharing and scientific exploration. We are thrilled to have with us Dr. Johannes and Dr. Christina, the head of MNS Society, and I'll give her a few minutes to give us an introduction about the MNS and the activities, and then we will proceed. Christina, how are you?
Hello, thank you very much. Thank you for taking this initiative and for organizing this first series of webinars for MNS. It's the first time that we are doing something like that in such a professional way, and also with in collaboration with the research networks from Boston, and we really thank them about that. And we thank also Dr. Luisa Pinto for making this collaboration happening. I would just like to remind you that the Mediterranean Neuroscience Society, it's a society for the countries of Mediterranean, but also beyond Mediterranean. And we're very open, and we would like to have more participants from different countries. Our goals and objectives are to strengthen the exchanges between Mediterranean neuroscientists, to promote education in the neuroscience area, and also to increase public awareness. So this activity is part of our educational activities, but it's also a very good opportunity to exchange research ideas and making collaborations. Also, we are determined to sustain scientific training and networking events in the Mediterranean area. And next year, around this time of the year, beginning of June 7th, we'll have our meeting in Greece. We'll let you know soon exactly the location and the venue. But the Mediterranean Neuroscience Society meeting of 2025, it will be in Greece, and we hope that you will all make it there. I just want to say that this webinar is recorded. So, and then at the end, you will have the opportunity to ask questions and interact with our speaker, Dr. Ioannis Sotiropoulos, who is, you will see, an amazing speaker, an extraordinary colleague, and also we also collaborate. So I cannot say too many good things, but you will see that it's an honor for us to have him as the first speaker. And then we'll have more in the next months. Yeah. So I'll give back to Dr. Zakaria the microphone to introduce him. Yes.
Yeah. Thanks a lot, Christina, for this wonderful introduction. And I'd like to say that our speaker today is Dr. John Ioannis Sotiropoulos. He's a renowned expert in neuroscience. Dr. Ioannis is a researcher CE group leader at the Institute of Biosciences and Application in the NCSR Demetritos in Greece. And he is an affiliated researcher at the ICVS Institute at the Medical School of University of Minho in Portugal. His research work focuses on understanding the orchestrating role of environmental risk factors on the onset of Alzheimer's disease, and with a special focus on the relationship between Alzheimer's and depression. Dr. Ioannis' innovative work aims to clarify the diverse factors that regulate the cellular role of tau as a key molecule of neuroplasticity and neuropathology. And he previously trained and worked at the Max Planck Institute of Psychiatry in Germany, at Columbia University in the USA, Riken Brain Science Institute in Japan, and MRC Center for Synaptic Plasticity in UK. Dr. Ioannis has received different prizes and awards, just to name a few, like the Janssen Innovation Award in 2017, and the Alzheimer's Association Award for Best Mentor in Neuroscience in 2021. Dr. Ioannis is an active member of the ELANIC Initiative Against Alzheimer's Disease, a network of basic and clinical scientists promoting translational and clinical research, as well as Alzheimer's disease daycare centers in Greece. Dr. Ioannis, we are very, very happy to have you today with us. Thanks a lot for this upcoming for this collaboration, and we are honored to have you with us to share your insights on a hot topic entitled Chronic Stress and Exosomes in Progression and Diagnosis of Alzheimer's Disease. Just before we give you the mic, and we will be all ears to your interesting session, just would like to remind the attendees that the webinar is being recorded, and at the end of this session, we will leave about 10 minutes for Q&As. Please, Dr. Ioannis is encouraging everyone to open their mics at the end and share their insights and opinions and feedbacks, and we will discuss as many as possible. And also in the chat box, we will share the link for an e-certificate and the link for a feedback survey, so we get sure we keep updated about your opinions and insights for future webinars. So without further ado, let's dive into today's topic, and please join me in welcoming Dr. Ioannis. How are you?
Thank you. I'm fine. It is a pleasure to be here. I think, as Christina said, and as you said, I think one of the main tasks of MNS, of the Mediterranean Neuroscience Society, is to bring scientists, especially young scientists, together from different countries, from both sides of the Mediterranean Sea, from south and north, to interact and visit each other and learn from each other. And I think the first, I'm very happy to be the first in this series of seminars that Mediterranean Neuroscience Society starts in order to help education and interaction. Because as I told you, it's good to learn, but also it's good, especially for young students, we highly encourage and we will try to find the funding scheme to support them from different students from all around the countries that are participating in Mediterranean Neuroscience Society to visit labs and do part of their works, of their masters, of their PhD. And that's very important. Already France has already started a Mediterranean master program, and I'm very happy to hear that. I think more countries from both sides should start and join this effort, or should start their own master programs, because master is easier to handle, as well as exchange more PhD students. I'm sure MNS will find, thanks to Christina and the president of the MNS that supported, and all the scientific council, including the ex-president of Giovanni, that he did a great work. I'm now part of the council, and I think we should support and find the correct funding to help students from different countries of Mediterranean to go in other labs and learn and do parts of their research there. That's very, very important. So having this in mind, I want all participants to make questions, to discuss with us, and you are always welcome to email me. You will have my email, the first slide, but also contact me to come in my lab or Christina's lab or other people's lab, because science has to be shared. And the main reasons, and remembering Nora Abru's words, one of the founders of the MNS, of the Mediterranean Neuroscience, is we need to support each other. And despite the geographical differences, there's nothing else to separate us, just the sea. And the sea, as you know, and I'm Greek, so the sea was, you know, the boat to exchange culture and goods for millions or thousands of years. So let's use this platform and MNS to exchange science and invite students to come in different labs, in our labs. I'm volunteer the first two people to join my lab and do for a few months their work in my lab. And so Christina and others has also said, I know Luisa from Portugal said the same, I know other people from Italy offer the same. So I think that's important. So please guys, try to join. I'm here from the Institute of Biosciences in Greece, as well as for the ICVS Institute in Portugal. So as you will see, we have created a nice family, a nice scientific research family where we work together and we work better and more productive together. So don't afraid to work with each other. I will give you an example today with the work I'm going to present, that the majority of that is produced in this bridge between ICVS Institute in the Medical School of Portugal and Democritus Institute For example, many students from Greek students from the lab in Greece are now in ACVS under the coordination of Dr. Joanna Silva, which is also part of the MNS, and Luisa Pinto, that is also the General Secretary of the MNS, and vice versa. There are Portuguese students that visited my lab in Greece, and I think this should extend with both sides of Mediterranean. So without saying more, I want to share to start. Let's share the window, see how it will go. And can you see my slides or no?
Not yet.
Not yet.
Can you see it now?
No, before we could, when you tried before, but now not yet.
Okay, let me go
I think
back.
there is an arrow. There is a button with an arrow. If you can press on the present now, maybe you can share your screen.
Can't share your screen. I will probably share the whole screen.
Okay.
Allow to share screen. Share the screen. Can you see now? I think yes.
Yes.
Yes, now
it's
something
coming.
is happening.
Is it coming now? Can you see the
Yes.
full
Yes, we can
screen?
see
The
it.
full screen? All right. So this is a very sunny day. The photo is a very sunny day from Democritos Research Center that includes 5 institutes. I'm in the Institute of Bioscience. My lab is called ExoBrain, Laboratory of Brain Exosomes and Pathology. It is in Athens. And as well, I'm affiliated research, as I told you, the ICVS Institute in the Medical School of Minho in Portugal. So both Mediterranean countries working together. Very nice example. Here is my email, which you can also see it. So as you see in this slide, you'll see that we have the progress of biomedicine that has managed to increase lifespan around the world, including both Europe, Africa, Asia, and America. And this increase of lifespan has a lot of benefits for all of us. And you can see, for example, different kinds of diseases are reduced by time. And this is data of the last 20 years, for example, like stroke or HIV, all these deaths are decreased. However, we have a big increase of diseases of the brain, such as Alzheimer's disease. And indeed, this is one of the next pandemia, we call it, in a way that we're expecting that the Alzheimer's disease cases, the people that will suffer from Alzheimer's disease all around the world will triplicate it up to 2050. So what is Alzheimer's disease for the people that they don't really know? It's an age-related neurodegenerative disorder that affects the brain mainly. And it comes clinically, it's manifested with memory loss and mood and personality deficits. And one of the main characteristics of that, let me do a laser pointing. As you can see here, we have a big atrophy of the brain that it indeed is based on the atrophy of the neurons and the loss of spines. As you know, spines is a major structural and functional basis of memory in our brain. So what we know since Alois Alzheimer, that really described 100 years ago this disease, he described also the two main histopathological hallmarks of the brain of Alzheimer's disease patients. One is the amyloid beta plaques, which is deposits, accumulation of a small peptide called alpha beta or amyloid beta, and neurofibrillary tangles, which is indeed aggregations of a cytoskeletal protein, and this is inside the soma and the dendrites and the action of the neurons. So this small peptide, the amyloid beta, is a small part of a bigger protein called amyloid precursor protein that it is cleaved, and then it is released outside the neurons, accumulated in this format that I show you with my screen here. Now, tau, as I told you, is a cytoskeletal protein that is mainly found in the axons and very little in the soma and the dendrites. This is a neuron of a healthy brain, and tau binds to the microtubules. So this is the microtubules of the axon and stabilize them. However, in pathological conditions such as Alzheimer's disease, we have an hyperphosphorylation, an abnormal hyperphosphorylation of tau that indeed detaches from the microtubules, it aggregates, and then we have a problem with cytoskeleton. That's why we have this atrophy So we lose dendrites and spines. Even more, tau is accumulated now to the back part of the neuron in the soma, and that creates a lot of toxic events, such as damaging the intracellular trafficking of the neuron, such as problems with mitochondria and many others. But what I want you to see is that this amyloid beta is in high concentrations, because we all have amyloid beta inside our brain and inside our body, but it's very low production. What happened in Alzheimer's disease, this amyloid beta you can see here is 10 times more than usually in Alzheimer's disease brain. And this amyloid beta in high concentrations, it is toxic and damages different structural and functional points of the brain, such as the molecules, such as interacting with synapses, or damaging neurons, or even local circuits, neurons together with microglia or astrocytes. And this indeed affects different networks of our brain before we have the final problem of memory or mood deficits that characterize the disease. But what I would like to say here is that recent evidence show that these toxic effects of amyloid beta in the brain is basically mediated by tau protein, and we will see how. And how they did that, they used some animals that they could remove tau, and they could block a vast majority of the neurotoxic actions of amyloid beta in the brain. So it seems that Alzheimer's disease is a disconnection problem, because you lose contacts and communication between neurons and molecules and different brain areas. And this toxic effect is mediated by tau protein. Now, another very, very important thing that I would like to highlight from the very beginning is that, as you can see in this scheme, if this is the point, this is a timeline, okay? The disease starts here and it progresses. However, we have the first symptoms just right now in the first red triangle. Then later we have diagnosis, and later we have the death of the person. But look at this. This is the clinical phase. So from the time I have some symptoms and go to my doctor, then we will have all the rest. But there is a long preclinical phase where the damage in the brain, the production of this amyloid beta and amyloid beta plaques, the tau hyperphosphorylation, the neuronal atrophy and the synaptic loss that happens many years before the first symptoms. In other words, there are studies, as we see later, that they claim that even 15 or 20 years before the onset of the first symptoms, the first memory problems that will lead me to the doctor, even 20 or 15 years before that symptoms appears, we have a damage in the brain that has already started. So the lesions has already started many years before. Now, if we go to see, there are different therapeutic trials targeting either amyloid beta or tau or the inflammation that we know that it comes together. And this is a scheme from a few years ago where you can see the different drugs that are designed to stop the disease, either when they target to stop the amyloid beta, or to stop the tau, or to stop the inflammation, or different targets that are happening inside the Alzheimer's disease brain. And you can see that this is, for example, 50 or more than 50 agents in clinical phase 2. Because as you know, when we create a drug, we need first a lot of animal or toxicity checks before we enter to test it in humans. And that's a long process. And there is different clinical phase 1, 2, and 3, and later on. So, but you see, for example, that only a few of them pass in the clinical phase 3. But even worse, if half of them, they pass to clinical phase 3, as you can see here, in the last 10 or 20 years, how many new drugs for Alzheimer's disease you know? One, and we are not sure, this amyloid beta antibody that appears in the States just last year. But still, 99% of the clinical trials failed. And the main big question is, why, guys? Why this is happening? Are all scientists working in Alzheimer's disease field stupid? No. Is it because we don't put a lot of effort and money? No. So what I want to discuss and present with you today is, I think we see only one parameter of a very complex tree, of a very complex disease. And here is where the new era of medicine, the precision medicine, enters, trying to suggest a more, let's say, holistic approach. This means holistic is a word that tries to include all the different parameters, the whole. And in this scheme, for example, you see that despite the different damages or molecular or cellular lesions that happen inside our brain related to tau or amyloid beta increased productions inside the brain, this results on a synaptic dysregulation in different regions and brain networks before we come to the cognition. But in order for this to happen, there is a lot of parameters that can stop, but attenuate this damage in the synapse, in the memories, and in the different brain areas, or they can accelerate. As I told you, from the first time we have this molecular change in the brain related to Alzheimer's disease, up to the first time I have the first behavior you can see here, it can be 20 years. So this is a big time window, and indeed it's a window of opportunity where we can attenuate, we can really slow down and stop this process. How? We know there are different parameters, for example, physical activity. You all know that we have the production of neurotrophins, as well as improvement of cardiovascular function that really helps the brain and reduce all this damage that amyloid beta and tau is doing. Then we have the Mediterranean diet. So don't use too much processed food or sleep well. And on the other hand, there are parameters such as the chronic stress, chronic psychological stress, which I work and Christina and others work on, or the problem with the insulin regulation or inflammation or even cardiovascular disorders that they can really help and push further the damage of the brain that has started because of amyloid beta and tau. So in other words, precision medicine is trying to see all the problem and not just the molecular part of that. So what is now precision medicine trying to do is to move away from the old-fashioned, old way of medicine, which we call it in English or American way, I would say, one-size-fits-all medicine. This means what? This means that here, let's focus to understand a bit this scheme. So these are different colors of people. These are all Alzheimer's disease patients. But indeed, probably what we call Alzheimer's disease, it's not one disease, or it has different subtypes of the disease. So some people in their brain, they have a lot of inflammation, and they start with a lot of inflammation, and then the amyloid beta and tau comes. Some others, they have a lot of amyloid beta, and later they come with tau. For example, some patients start with motor problems. They cannot move well, for example, where some others start with depression, and then later it comes the memory deficits. However, up to now, this old-fashioned way of treating patients in medicine, and in Alzheimer's disease particularly, since we focus on Alzheimer's disease today, it treats all these patients with the same drug. And some, they can benefit, some they have no benefit, or some others, they have even, diverse effects, like side effects. So what the precision medicine is trying to do is to classify these patients on different subtypes of that they refer to their pathology in the brain. How it's going to do this? How we do that stuff? We take into account the individual characteristics of each patient, like the lifestyle, the health history, if they're living, for example, in an area with air pollution, as well as individual multi-omic data from each of us. So I will give, for example, my blood, and through my blood, they will have a proteomic profile, metabolomic profile of my own body, of my own cells. And then they're going to classify me in categories, in these different categories. And then they will give me the correct treatment in the correct time. In other words, in Alzheimer's disease case, we can have different subtypes of diseases, and based on which subtype I belong, I will receive a different treatment, not a different drug. Some years ago, precision medicine was called personalized medicine, but this term is not anymore correct. Why? Because people think that, you know, I'm going to personalize and make a drug for me. No, that's wrong. So what we're going to do is we're going to really understand as carefully as possible the molecular profile of the disease state I belong. In other words, United States of America and NIH guidelines for Alzheimer's disease diagnosis is as below. Please forget the clinical symptoms. Go back and use more of the molecular data, the individual molecular data of each patient in order to understand what is the subtype, how they suffer, and how you should treat. So in order to have a very good understanding, I gave you an example with blood. We need better biomarkers. In this scheme, you will see, for example, that if I have good biomarkers, and this is the decline of cognition and function we all have in our life, this is a healthy aging. You all know that as we age, we have a small decline of our cognition, of our function. What happens in Alzheimer's disease? We have this red line, very big and sharp drop that it starts. And here, for example, is the prodromal state of Alzheimer's disease, which is called mild cognitive impairment. So if I have good biomarkers that I can understand, biomarkers, in other words, is called biological markers, to understand what happened inside my brain. What I can do, for example, yes, I can understand here when it starts early, the disease, not here when I cannot do anything. Because it is very important to, as you know, for everything in medicine, is to start treatment as early as possible. So if I have good biomarkers, we have early diagnosis or even prognosis. So in that way, I can understand before I have the first symptoms that something is wrong in my brain. And it's a good opportunity because I can have primary prevention intervention. For example, if I know now that in my brain, I start have tau accumulation, I will start have exercise, I will start change my treatment. So I can do things that can delay it since there's no drugs. And if I have good biomarkers, I can also understand what happens inside my brain if I use a new drug, but I'm not sure if it's working. So we need better biomarkers. And this is where the main focus of this talk is based, is the exosomes. So what is exosomes? Exosomes are very small extracellular vesicles. You can see here, for example, they're secreted by all the cells. In our case, we're speaking about brain cells, for example. For example, this is a typical neuron that it has accumulated tau inside that. It's a diseased neuron. So what is believed that exosome, indeed, a diseased neuron is secreting exosomes that includes protein, microRNA, DNA from this cell, from the donor cell, to the next cell. This cell is picking it, either directly or through microglia, in order to become disease. And so first thing that we should know for exosomes is that probably they transport the disease from one neuron to the other. And just to tell you that exosomes are secreted, it's part of the endolysosomal pathway. This is a pathway of degrading proteins. And instead of going to lysosome for degradation, the proteins that were not used anymore, for example, like the old tires, that after we use them, we need to degrade. Instead of degrade them, we can also secrete it outside the neurons. And these are called exosomes. Very simple, okay? I don't want to say different ways of secretion, but I want here to focus that you see that when the exosomes are received by the green, this postsynaptic cell, there is a lot of things that can happen. And because they transfer DNA, protein, or microRNA, they can have a lot of effect on the cell response. Here we have a very nice review from last year when you can see that there is a lot of function from exosomes. I don't want to stay too much because there's no time. You can see this in this paper, which I think it's open access, the experiment neurology. If you don't have it, please email me. I will be happy to send you. When you can see a lot about things, about the different functions of exosomes in communication between cells. And very, very importantly, not only exosomes secreted by neurons or glia cells, they transport the disease from one area, brain area, to the other, or from one neuron to the other. But they also, because they're small, they cross the blood-brain barrier, and we can detect them in the blood. Oops. I think I said the magic word, so the blood. So which biological fluid you think it's easier to collect? I'm sure it's the blood, because we can take in every place, in every small village, in every small hospital, or every doctor, even not with a medical unit. You don't need a hospital to just take blood. And there, you can, if you can isolate, and this is what I'm going to show you today, we isolate exosomes coming from the brain, from the neurons, of the glia, and in that way, we can have good biomarkers, or at least this is what we believe, that we can have good biomarkers for what happens inside the brain. And this is exactly what I'm trying to say to you. From brain pathology, the brain secretes their exosomes or extracellular vesicles, to be absolutely correct. But I don't want to go to details for now. And these brain exosomes, or EVs, we're collecting them in the peripheral blood. So the idea is that we have brain pathology, we collect the brain EVs in the peripheral blood, and this can help in more reliable diagnosis or even prognosis. Okay? Now, the difficulties working with brain is tissue in order to understand what are these brain EVs and what they transfer. Because if I know that the brain EVs from Parkinson's disease, for example, and from Alzheimer's disease, from any kind of disease, they transfer this type of proteins, then I will go in the blood, exactly here, I will go to the blood and collect. So we focus in order to understand on characterizing the brain EVs. The problem is working with brain EVs is that most of the times all the techniques digest the tissue. So in that way they break the cells, and this results in a lot of dirty. So without giving you too much, in ICVS, we have developed a new protocol, a new method actually for isolation, where we allow the tissue or the cells from patients, and this works for both human and animal tissue, to secrete spontaneously the exosomes. And this is in collaboration, we have developed this in collaboration with Bruno Costa Silva in San Paligo, also with Dimitris Kapogiannis, John Hopkins, as well with Clarissa Waits in our lab by a very talented ex-student, MD PhD student of mine, Patricia Gomez, which is now she's doing her specialization psychiatrist, and now she's indeed in with Dimitris Kapogiannis and John Hopkins in Baltimore. That's a bit briefly how. What I want to show you is that our technique, we use different techniques to see, like cryo-electron microscopy or TEM negative staining, and we see that our release or nano-tracking particle analysis, we see that our method, which is shown with red, creates more pure exosomes, more clear exosomes in the and less bigger vesicles that are not exosomes. And we also did proteomic analysis, I told you, and we find a lot of neuronal. I don't want to go to details. I just show you proteomic analysis, which we do in the lab, and we see, for example, that our exosomes, our method, give us more pure exosomes that include a lot of neuronal proteins, but also microglia, astrocytes. And indeed, this is true, because not only neurons in our brain secrete exosomes, but also other cells. And we did also a complex nanoflow cytometry. What we saw there is that we see that our method is more advanced in a marker of small EVs, which is very, very important because that shows us that it's more pure. And then we give another technique, the exoview, in order to see what it is inside. And we find, for example, that there is tau inside these exosomes, a protein that we know that these exosomes should transfer. Then we did another experiment. I don't want to say details, but this is very important. In order to check the functionality of the exosomes that our method is going, and this is, as I told you, exosomes are secreted from one neuron, and then they are picked by the dendrites or the soma of the other, and then they are moving. So this is exactly what we did. We label with a red dye, a special dye, these exosomes. We inject in one area of the brain, and then we try to see if they have moved in the other area, which is synaptically connected with these neurons. And this is exactly what we see. We see, for example, we inject it here, and then we see they move down to the other area of hippocampus. I don't go to details because that's not important. Showing exactly this transportation of exosomes and the cargo, all the proteins, the microRNA, they transfer, suggesting again that our method is working. And as I told you, not only exosomes are moving between tissue and different neurons, but also we can detect in the blood. And I'm going to share you an idea that we use in a protocol we use in the lab, where we can really isolate in our blood only exosomes from the brain. Why? Because in our blood, there is exosomes from different tissues, from our muscles, from our heart, from our liver, because exosomes are secreted from cells in order to communicate between each other. So what we need here, we use one molecule that it is expressed only in neuronal. I mean, there are more molecules now and more specific, let's say. And what we did using this technique developed in Dimitris Kapoyan's lab, and now we have and use it in the lab, we'll isolate and we, let's say, immunoprecipitate only the brain-derived exosomes. And here I'm going to give you some examples from different papers where you can see, for example, that just by using exosomes, blood exosomes, or brain exosomes collected in the peripheral blood, so no CSF, no brain tissue, nothing. They could just brain-derived exosomes in the blood of these patients, they could separate based on the level of tau and phosphotau and amyloid beta that these exosomes had inside their cargoes. In other words, just by using and measuring what these exosomes coming from the brain and collected in the peripheral blood, you can understand if this is a patient or these patients, you can classify these patients if it is healthy or Alzheimer's disease. For sure, this is not something that we use in the clinical practice because it's still under development, but it is very promising that based on, and this is not only for Alzheimer's disease, for other diseases, and not only for brain diseases. This could be true. We can have, for example, some biomarkers for cardiovascular disease or other type of diseases. Another example in this paper from Dimitris Kapoyan's lab is where they could predict, based on the brain exosomes collected in the peripheral blood, they could predict, this is a longitudinal study, okay, that happens over 40 years, they could predict the cognitive decline later in life. So if these people, in other words, if these people has higher phosphotau or total tau in their exosomes in their blood, they could see that their cognitive decline by time was bigger than the ones that they have lower tau levels. And this is just an example saying that it's very promising even for prognostic reasons to use brain exosomes that we collect in the peripheral blood. And here is again from a review we wrote where you can see, for example, that as we have a progression of the Alzheimer's disease from preclinical to mild cognitive impairment and later to really established Alzheimer's disease brain pathology, there are two things that we can collect changed, or two things so far. Inside the exosomes that we collect in the blood of AD patients, we have increased phospho-tau and amyloid beta levels. This is exactly what happens in their brain. In Alzheimer's disease patients, as I told you, we have increased amyloid beta levels, increased phospho-tau or total tau levels, and look, in their exosomes, we detect exactly the same. If you compare exosomes from Alzheimer's disease patients from healthy ones, you have increased levels inside the exosomes, okay? So, and vice versa, you have reduction of synaptic proteins inside the exosomes. What happened in the brain, do you remember? We have a lot of atrophy from neurons and loss of synapse. Loss of synapse means reduced synaptic proteins. So I gave you a nice example, for example, that this, based on the levels of the cargo of proteins or microRNAs, that you can see more details in this review, that we can see exosomes can show us what happens inside the brain. In other words, brain-derived exosomes in the peripheral biomarker, in the peripheral blood, can mirror what happens inside our brain. That's the hope, and there is a lot of disease. And in the last part of my talk, and I'm finishing, I want to show you that the work we have been developing, not only with exosomes, but also with the chronic stress, because chronic stress, it is an accelerator of the disease. It is a risk factor now. It's very, very well accepted. Previous work from us have shown that if you expose animals to chronic stress, you can aggravate tau pathology, or you can really start depression by increasing amyloid beta and phospho-tau. That's a lot of different mechanisms we have described on different papers. That's some of them. For example, where we have the tau phosphorylation and the burn of the synapse, or the problem in degradation mechanism, or problem in mitochondria, or even problem with neurogenesis. And these related with the phosphorylation of tau and the accumulation of toxic species of tau inside the brain that is triggered by chronic stress and the exposure to stressful conditions. And indeed, what we did believe that probably there are common neurobiological bases between depression and Alzheimer's disease, because we know very well that depression is a risk factor for Alzheimer's disease, increases the risk for Alzheimer's disease. And indeed, what we have been suggesting for the last 23, 24 years since I started my working on the field in Max Planck Institute of Psychiatry in Munich is that probably this decline we have by age in our mood is triggered more when you have more and more chronic stress or higher levels of chronic stress. Then this, through the depression, it goes to the Alzheimer through common neurobiological mechanisms that they trigger neuronal function atrophy. You can read these papers and you know more. But I want finishing to show you how chronic stress and how the stress hormones, the glucocorticoids, affect neuronal mechanisms that accumulate toxic species of tau inside the neurons, and this results in neurodegeneration such as Alzheimer's disease. As you know, all the proteins are degraded through ubiquitin proteasome system, but in disease conditions or under chronic stress, as I will show you very, very briefly in the next two slides before I stop, we also see that proteins are degraded through the endolysosomal pathway. You remember this is the pathway that is involved in the biogenesis of exosomes. But also, another mechanism that is also activated under disease or stress conditions is the autophagy. You probably know about autophagy because the Japanese guy who invented won the Nobel Prize some years ago. So just making many years of work in one, I want to show you that chronic stress inhibits both the endolysosomal pathway that leads to the accumulation of tau protein inside the neurons, as well as the autophagic pathway that again leads to reduced degradation of proteins in the lysosome. Here is the two papers. I don't want to more, and there are more after that. That was the original, but there are more now. But as I told you, this endolysosomal pathway is also involved in the exocytosis of exosomes. So we recently saw that chronic stress, it induces mood and memory deficits, it induces atrophy, but also it releases a lot of exosomes that they carry proteins that they can predict what happens inside the brain. Here is a paper and the process from Patricia Gomez and the people, and trying to show you the multi-phases of chronic stress damage on the brain. I want to finish with this slide, showing that chronic stress and Alzheimer's disease share a lot of common mechanisms that they damage synaptic plasticity and neuronal, leading to neuronal atrophy and synaptic malfunction, leading to memory deficits and depressive pathology. And I want just one minute of you. You can go in our website, which is the Hellenic Initiative Against Alzheimer's Disease. So it's a bunch of different researchers, clinical, basic, as well as people working with patients from different parts of Greece, as well as from abroad. For example, Professor Kostas Liketsos is professor at Johns Hopkins University in the United States, as well as the director of Alzheimer's disease. So the Hellenic Initiative Against Alzheimer's Disease, it's called Hellenic, but indeed we're trying to help and support research, basic translational research, as well as the daycare centers for Alzheimer's disease in Greece, but also abroad. So, and do research, we do dissemination, but also we have a lot of seminars once per month where you can see different people from Greece and abroad speaking about. It's free. You can just register and participate and make questions the same way, where you can see clinical and basic researchers speaking together. But we also have different books. The first one, it's for the general audience. The second is for medics and nurses. The first one, it's me and Christina, Christina Dalla, the president of the MNS, that we wrote together. And with that, I want to thank both the labs in Athens, in the Institute of Biosciences, as well as in ICVS Institute, in the Medical School of Minio, that together we're trying to work together for many years. And I think we can fight it better, this disease called Alzheimer's disease, and as well as all our collaborators. So thank you so much for your attention. I hope I gave you a brief and easy to follow idea about Alzheimer's disease and the different ways we target in the lab, as well as the different, let's say, approaches that together we can do if you want to join my lab or the lab in Portugal, or different members that are collaborating, such as Christina Dalla and the others. Thank you so much.
Thanks a lot. Thanks a lot, Dr. Ioannis. This is indeed a very interesting topic, and I think it's inspirational to have this new method for isolation of the exosomes, because it's a hot topic, and the point that it is it introduces a new method for more specificity. I think it's very interesting.
Thank you, Nermeen. So, is there any questions from the audience?
Yeah, attendees are encouraged if they can raise their hands, or
we can allow the people to speak and tell their questions.
So,
is it possible?
I think it is.
Yes, I'm allowing everyone now to raise the questions or type their questions, or they can also unmute their microphone and ask directly.
In the meantime, I can say again that this is an initiation from Mediterranean Neuroscience Society. Like different people of us that they're in the governing council, such as Christina Dalla, who is the president, but also Luisa Pinto, who is the general secretary. We're really, and Nermeen, we're really supporting this new series in order to interact and give the opportunity to more young students to learn and speak between them. I would also like to say that there is the MNS Young Scientists Network, that you can, it's participation. There's a lot of PhD students, master students, and postdocs that participate, so you can benefit a lot. We have a lot of fellowships for you to travel, not only in our meeting in Mediterranean Neuroscience meeting, but also in others, as well as we also want to give the opportunity from different people from both sides of the Mediterranean to visit labs. For example, you are very free, and we will happy to welcome you in my lab to do things either in Greece or in Portugal together with Joana Silva, which is a young group leader there, and we collaborate as I show you in the photo of the last slide. We are a family. Together with Luisa Pinto, which is General Secretary, we really encourage this. And I know Luisa also wants to receive students in her lab. So go speak with people, and we will find the funding tools. Soon, MNS will have a funding system to do this. We are working on that. Okay, so questions?
Yeah. Anybody wants to ask a question now? You can just submit it in the Q&A, or just raise your hand and open your mic and say.
And speak.
Yeah.
So I think we don't have
also
questions. Oh,
Yes, I want to thank you, Yanni, Dr. Sotiropoulou, and Dr. Zakaria. And if
Thanks.
someone wants to say something, anything, or want to ask something related to the talk or something else, this is a good opportunity. Of course, you can always email us as well. Thank you very much for joining, and we hope we'll see you next time as well.
Thanks a lot. Okay. Johannes, I have a question about the your paper. I've just read your paper about the spontaneous release method for the isolation of the exosomes, and yeah, and it seems very interesting. What about the bicrotoxin treatment? Is it a way to make it more inductive to produce more exosomes or more specific exosomes?
Yes, indeed. This is based on the inhibition of inhibitory neurons that they, let's say, stop the neurons. The other way we do it with stress. You see, with stress, we have an activation of the neurons. So both are inducing. So you can either use we did both ways. So we use a pharmacological approach to increase the release of exosomes. And we try to see how sensitive is our technique. And also we block it. But also we did activation of neurons and increase by chronic stress, which we knew for our first experiment that this is happening. What it is very promising with this technique is that it can be used not only with brain tissue, but with muscular tissue. So it can be used, and it's very simple, it's standardized, and it can work both in human tissue as well as in non-experimental. So in that way, it can help us from the tissue pathology to the tissue EVs, and then these EVs that we will collect in the third level in the peripheral blood. That's very important because we can have an understanding of what is happening, a molecular, let's say, footprint, a molecular signature of the damage of the tissue through the EVs. That's the main idea.
Yeah. And have you tried to track these changes in the exosome release or the constituents of the exosomes according to the stages of Alzheimer's in AD patients? Or,
yes, there's a lot of work. There is the work from us. We focus more on chronic stress, that we do the same in AD patients, but there are also from others, not from chronic stress. We are doing that, but there are other studies, a lot of studies from Alzheimer's disease and other brain diseases, MNS, ALS, that they are trying to understand the footprint of the brain lesion over the progression inside the biomarkers. If you want my opinion, I think in a few years, 3 to 4, 5 years, we'll have a panel of proteins and microRNAs that we will help diagnose of Alzheimer's disease. Based on exosomes and what they carry, or in general based on blood. So blood is a key biological fluid for diagnosis nowadays, and I'm sure as the technology advances and we are becoming, you know, when I was a PhD student, me and Christina or Luisa, Luisa Pinto from Portugal, as I told you, which is the General Secretary, and she also facilitates us with the support from Elsevier, from the journal, the brain organoids, and thank you, Luisa, again for that. I know you are in the audience. So when we were students, we could never imagine that it's possible to have a single cell proteomic analysis. So we have a proteomic analysis of one cell. Today is possible.
Yeah.
It's cost a bit, but then in a few years it will be like, 50 euros.
Yeah.
As we have an advance of technical, advanced based on sensitivity, because we need sensitivity, because these are very, very small, okay? Very, very small nanoparticles, size of 150 nanometer. So we need sensitive techniques, and this is where the whole field is moving. More sensitive techniques in order to be used in a clinical practice in an easy set.
Yeah, indeed.
Still, we need a lot of work to go there, but this is where we go now.
Yeah. Okay. I think this is it for today, and again, we are very, very honored. Yeah, we're very honored to have you today with us, and looking forward to further meetings and maybe further discussions about. Thanks a lot, and thanks a lot for the lovely
Please
audience.
check our website.
Yeah.
Please check the website that just Christina sent it with MNS and the news and events. When you see things about new seminars that they will come, this is the first. As well as opportunities for fellowships that MNS is giving. For example, the previous president, Giuseppe, is organizing a dementia course for 1 week in CC in July. And MNS, as well as the course itself, provides travel fellowships that cover all the costs and everything for students from different countries to go there. Please apply, go there and apply. So check our website for more news.
It's at Catania.
It's
in Catania.
Yes.
And I put the link for the MNS website for the news, and we have everything there. And also we'll announce also some travel grants for the Eurogenesis meeting at Bordeaux.
Yeah.
So hopefully
Yes, which is also in June, more soon.
Yeah.
Yes.
There's also the feedback. Yeah, sorry, Christina.
The last thing that we'll announce that as well next month is that if you join MNS, then you will also be a member of FENS, the European Neuroscience Society. So you'll have the benefits of FENS as well, like webinars
and you can apply for travel grants for FENS. Yes, discount, and you also can apply for travel grants, which cover all your costs to go to FENS and other meetings. So there's a lot of tools supporting young scientists, which is free. Use it, guys. It's your time. Don't afraid to fly and go and visit places. Use these tools that we are trying to collect for you. You are the future. So just use it and go and learn life and learn some more techniques and some more science.
Great, thank you. I think we used all our time.
Yeah.
But we'll
Thanks
see
a lot.
you again soon.
Thanks.
Bye.
Thank you. Goodbye, guys.
Thank you. Thanks. Bye.
Have a nice
Bye.
afternoon. Goodbye. Ciao.
Yeah,
Ciao.
thanks a lot. Bye.